I'm Bill McShea. I'm a wildlife ecologist. Peter was talking about the future. My job is to talk about the past. I'm like the ghost of Christmas past. I'll be here almost 40 years. So Peter said, could you please cover the last 40 years? And you have 10 minutes. So I decided I can't do that. But what I can do is talk about myself. I can always talk about myself. But I can talk about how my science has transformed over the last 40 years to be more the conservation science that the center wants to be doing. So we're going to go through that process here of me. And I'm sorry for all the people I'm skipping. I'm sorry for all the things we're not talking about. I'm going to concentrate on the ecology here at the center. So I came here to study the star-nosed mole. There's the mole, and that's me. And it was totally for animal programs. Ed Gould wanted to have star-nosed mole display it downtown. Wanted someone to come who could keep those things alive in captivity and could study those rays. I agreed to do it. I could have been downtown. I could have been here. It was an animal program project. While I was doing this project, the center was wrapped up in this crisis of deer management. The deer were eating all our hay. We actually had a congressional meeting where they just lambasted Chris Wemmer on why you're trying to shoot the deer. Can't you do something about this rather than shooting them? And we started a deer ecology program, pretty much to understand why we have so many deer, where the deer are coming from, how can we manage these deer. It was focused on land management with respect to hay production. The big function of this center is still hay production. And at that time, it was, what are we going to do about the deer? So we were collaring deer, capturing deer, driving deer. It was a lot of fun. And I went away from the mole and started doing more deer stuff than I was doing mole stuff. Then we got a research coordinator, John Rapole, here in the corner, and he said, I don't care so much about deer. What else can you do? And I said, well, I can do rodents. I don't care about rodents. Well, I can do birds. Oh, I love birds. So we started a project that combined deer and birds and rodents and acorns, and it became a trophic interaction, food webs. For more than 15 years, we did quite extensive studies here in the center on how these animals all interact with each other. So while we're trying to manage the deer, how managing the deer impacts the migratory birds, how it impacts the rodents. Well, we actually found that predators were switching between the rodents and the birds depending on the acorn production the year before. And the acorn production depended on how many acorns were being eaten by the deer. So all that was related. It was a great study. I really, really enjoyed it, but it took us beyond looking at individual animals for management reasons to looking at ecosystems and the interactions between species. While that was going on, Chris Wemmer said, hey, we have an opportunity to work in Myanmar. And we have a deer here at the center, Eld's deer, and they have a deer in Myanmar that's having problems. Do we have anybody here who's a deer expert? And I said, hey, that's me. Okay, let's go to Myanmar and let's look at the deer there and let's see if anything there relates to our captive management and is anything that that we learn from the captive management relative to the deer in Myanmar. So we're still connected with animal programs, but we're getting more outside the center and more toward an international focus. And several things happened. That was a seminal project for the center in that Chris Wemmer, it was an all hands on board. You may recognize a couple of those pictures there, but we brought probably 15 different people from here out to Myanmar to study different aspects of this problem. We went from one researcher doing one project to really collaboration at a much larger scale we brought out geneticists and plant people and small mammal people, and we created an entire team. Because what we found out is that this wasn't really a deer problem. It was a people problem. And we had to find a way to engage the people, the local people in Myanmar around this one Chatin wildlife sanctuary. We built houses for those people. We set up camps for them. We made a plant team. We made a bird team. We made a deer team. We engaged the community in all these different activities. We supplemented the patrolling teams. We had to engage the community in order to conserve these deer. And it's, at the meantime, we said, well, let's do the trophic interactions. Let's look how these deer are interacting with the environment. And it became very similar to the situation here. These deer, the Eld's deer down there in the bottom, were dependent on this forest called Indang forest. That Indang forest was dependent on fire. You had to catch that forest on fire every year or the forest would decline. So it had to be burned. And the reason it had to be burned is because it generated all these grasses following the burn, and the deer needed those grasses. So you had to have the forest, you had to burn it, and the deer exploited those grasses that came up. The problem is that people messed up with that cycle. People removed the trees, people put their cattle in there to eat the grasses, and those human-wildlife conflicts broke that cycle that we were finding in that location. But it was a fascinating thing, and we spent a good 10 years there looking at this study of the people, the animals, and the forest. You can't talk about conservation here without talking about giant pandas. You know, we're all giant panda obsessed here at the zoo. And I want to give a shout out here to Dave Wilt in that we had captive pandas since the 1980s, but we were unable to get into the reserves in China until Dave Wilt and Joe Gale Howard opened up that whole situation by going around with their biomedical survey. Their biomedical survey of captive pandas got us access to a lot of reserves, a lot of connections, and we used those connections to start activity on the ecology of the giant pandas. And I would say that Eld's deer, none of you people know about Eld's deer. It's a very low profile animal, but giant pandas, now we're into politics. Now we're into high profile animals. We're into the government involvement. It's a much bigger situation for us to be in. And it was an important something for us to get involved with. So the way we started there was training courses. Melissa, myself, others offered training courses. And a lot of this, we should give a shout out to Rudy Rudran. He was already doing wildlife management courses throughout Asia, and we could modify the courses that he was doing, the network that he created, in order to tailor it for the needs of the Chinese community. And their needs were technology. They loved technology. If we could bring technology to their wildlife science, that's what they wanted. So we had a ton of courses. I think we trained in all of the reserves, all the giant panda reserves over the course of time, both in laboratory techniques and in field work. We used that training to start research projects. So like that's Barb Wolf up there in the corner, putting down a takin. So you put a radio collar on that takin, and we're doing takin ecology. We're collecting fecal samples to do hormone surveys for Asiatic black bears. We're doing interviews with people around the reserves on why do they kill bears or why do they not kill bears. All of that was technology that was brought into the situation in order to make this suitable for the Chinese. I'm a camera trap guy. I know we're obsessed about giant pandas, but there's a whole supporting cast around in these panda reserves. They don't only contain pandas, they contain a lot of wildlife. And we can use that wildlife to understand how well they're managing these panda reserves. So just a couple studies. There's a study on the left here using the camera trap data to look at the retention of 4 large carnivores in the giant panda reserves, where there's still these carnivores and where these carnivores are missing. Sadly, those giant panda reserves are not doing a good job with carnivores. Most of the large carnivores are disappearing because those panda reserves are not big enough for those carnivores. The other study was looking at corridors between reserves, using the camera traps to who's in those corridors, what animals are using them. Are all the corridors good for all animals, or are they just good for giant pandas? And it's just an example of things we can do based on our training courses. I've taken that now and I use that in the lessons I learned there and use that in all of my research right now. We have this, Peter mentioned this Snapshot USA thing. This is a continental scale thing where we're trapping in all 50 states. It's all by volunteers. Everyone is putting their cameras out at the same time in the same way in order to share the data. And we all use that data for conservation. So that's a project that really builds off of what we learned in Myanmar and China. And we have had the opportunity to set up new programs. And we're starting out these new programs using everything we learned before. So we're starting out these programs with science, grassland science. We're starting it out with community engagement, talking to the stakeholders, the ranchers, the Native Americans who are living in this area. How are they using wildlife? And then we're still connected with the animal programs in our reintroductions, reintroducing bison, swift fox, black-footed ferret. All of that brings everything we have together. So what have I learned over this time? I've learned that conservation takes a long time, and it has to happen at large spatial scales. My original master's was on a 1 hectare plot. I haven't worked on a 1 hectare plot in a long time. We have to involve research teams that come with all different kinds of expertise. We have to engage the local people, or what we're doing is essentially useless. And I tell my people all the time, conservation is a team sport. It's no longer any good for you to go and look at your little mole in the corner of the woods. I miss my moles, but that's not doing us much good as far as conservation go. So these are the lessons I've learned over this 40 years, and I think it fits well with the kind of conservation science you're going to hear from now. Now all our speakers are going to talk about what are we doing today rather than what we were doing 30, 40 years ago. So let's move on to our next talk. You're ready. I say the next talk, but the next is a song, Amanda Liew from the One Health program.
I think so. Hello, everyone. Let's see if I can, is it this button? It's this button. Okay. We're fine, right? I can do a balancing act. Yeah, I don't usually cause this much chaos, but hopefully everything makes sense by the end of this. Highway to One Health. What is One Health? A collaborative approach that brings together people from different fields, physicians, vets, and scientists everywhere. They share the load to care for animals, humans, and the earth. Our project aims to integrate conservation and public health. Zoonoses, yeah, there are so many definitions with shared animal and human conditions through the bite of a bat or a cranky rat or inhaling feces of a cat. Then there's stuff from ticks, mosquitoes, and fleas in the worlds of food and waterborne disease. There is still a lot that we don't know. Zoonotic host, how do you know it's true? Can this animal share with you nasty germs like fungi or some worms? Providing science for policy, bringing light for all to see that the world doesn't need a repeat of 2020. Wildlife trade, you never know what's in those crates. Snakeskin boots or some live primates, armadillos or shrimp, an endangered wildcat, an illegally smuggled giant pouched rat. They're all hosts of germs that could make people sick, things that could start another pandemic. there is still a lot we need to learn. Okay, you didn't think I was going to do the whole thing in a song, right? Thank you. So now that I've thrown some pretty lousy rhymes at you, let's get to the real content. What is the highway to One Health? So as you guys all know, the pandemic threw a lot of mix-ups into everything and everywhere. It really challenged scientists to think about things in ways that they never had to before. So this is kind of where it sets up the backdrop for our project, the One Health Protect team. So wildlife trade is a very big global industry worth over 100 billion annually, and the United States evidently contributes a lot to this large number. Over 100 million live animals. Keep in mind this is separate from animal products and parts, but just live animals are imported every year. Also, caveat, I'm not trying to say that COVID was a result of wildlife trade, but that when there are things happening at such high volume, it's difficult to regulate. So specifically in this context, there's limited disease surveillance, and you're probably wondering, or maybe not, why this is important. So zoonoses, which we're going to define as any infectious disease that a human can get from an animal, this is a really big worldwide burden. Over 2.5 billion cases and 2.7 million deaths annually. So to have just this giant thing looming over us and to not know how much of a risk it really is when you bring in something that could possibly infect humans, this is a pretty significant knowledge gap. So here at One Health Protect, our project is really aiming to possibly contribute to bridging this knowledge gap. We're basically answering this big picture question of which wildlife species have the potential to bring in these high consequence pathogens through the trade pathway that could be a really big issue in terms of human and public health. And the way that we did this was a twofold approach, making two big data sets. So the first data set is we gathered together a list of zoonotic pathogens that are listed as concern in the US trade, along with all of the reports of human disease that are associated with these zoonoses. And then the second part is we looked at the wildlife trade component by identifying any reported hosts through literature of these zoonotic pathogens from part one. And then we also collected and curated a data set from US Fish and Wildlife Services that reports all of the wildlife trade records from 2000 to 2015. And lastly, we have now stacked our 3 cars and we're compiling all of the information that we gathered through this process to do a risk assessment. And this is the ultimate product, is just a ranked list of kind of the dangerous or the scariest host species. And that product will be going to US Fish and Wildlife Services. Okay, gotta wrap up exactly how I started, right? Well, now you know about the fun we have working with each other globally, building teams, maintaining health security. So this is how and why we do the work here at the National Zoo. Keep wildlife safe and keep germs out. That's what the highway to One Health is all about.
You can download that on Spotify. Okay, our next talk is Katherine Mertes from the Conservation Ecology Center, talking on Sahara antelopes.
Hello, everyone. Thank you for the opportunity to speak with you at this celebration of 50 years of conservation impact by SCBI. The only way I can possibly follow an original live song is by telling you about a conservation success story that SCBI has been involved with from the very beginning. I'm largely going to focus on the species on the left, the scimitar horned oryx. Or before 1900, herds of thousands of oryx were seen across the Sahel region of Africa. However, due to increased hunting pressure, increased populations of people and livestock, and persistent civil conflict, the species declined in one range state after another. The species was last confirmed seen in the wild in the 1980s and was officially designated extinct in the wild in 1999. I'm going to mention these two other species here. Addax and damogazelle are two additional species of Sahelo-Saharan antelope. They are currently classified as critically endangered with less than 100 individuals remaining in the wild. And almost as soon as the scimitar horned oryx was designated extinct in the wild, work began to restore the species, and SCBI was a part of this work from the very beginning, with researchers conducting field surveys, veterinarians and keepers offering their expertise, scientists developing translocation and monitoring protocols, and institutional leaders convening conservation planning workshops across North Africa and the Arabian Peninsula. These efforts reached a milestone in 2016 when the first 21 oryx were released into the reserve fauna Wadi Wime, Wadi Hashim in central Chad. And because of SCBI's work and emphasis on post-release monitoring, nearly all of these individuals carried GPS collars. And that emphasis continues to this day, such that more than 90% of all oryx reintroduced in Chad have carried at least one tracking device. And we've used this monitoring data to assess conservation progress when we combine field monitoring, animal tracking, and ground transect data to estimate oryx survivorship. We obtain estimates of more than 80% for females and more than 75% for males. And when we combine survivorship with other metrics, we obtain extremely promising estimates of population size and growth. We've also used animal tracking data to understand how reintroduced oryx are behaving in the extremely seasonal environment of the Sahel, which is completely different from their background in captive institutions. And when we calculate how much time oryx spend performing different behaviors across seasons, we see that they're responding appropriately to seasonal cues, spending more time foraging and traveling during the short, intense rainy season, and more time resting during the hot, dry season when temperatures can reach 120 degrees in the middle of the day. Based on the growth and behavior of the reintroduced oryx population, the species was downlisted from extinct in the wild to endangered in 2023. This is an incredible conservation achievement, again, that SCBI was involved with from the very, very beginning, planting the seeds with conservation planning, nurturing those developing populations with applied conservation science, and advocating at every step for the post-release monitoring that made this downlisting possible. And we've leveraged this conservation success to add the two Sahelo-Saharan antelope I mentioned earlier to our reintroduction efforts. Since 2020, more than 115 addax have been released into the reserve in Chad, and more than 150 calves have been born in the wild. In 2024, we released our very first group of damogazelle into the wild. And because of this emphasis on post-release monitoring and animal tracking, these animals not only add numbers and genetic diversity to the local Dama population, but they increase our capacity to monitor this small, declining, and poorly understood population. Every day, we work with our partners to apply the lessons learned from oryx to all three species. And as we meet here today, there are 14 collared addax in acclimation pens in the reserve in Chad that will be released in a few weeks, and a second group of damogazelle that will be released later this year. A quick thanks, as always, to the many organizations and people without whom these reintroduction activities are simply not possible. That includes all of you, and that includes many people and organizations across the entire world. Thank you for your time, and if you have any questions or would like to discuss any of these topics further, please feel free to reach out over email or find me later today.
Our next talk is Kristina Anderson-Teixeira, going to talk on the ForestGEO program.
Okay. Good morning, everyone. It's so nice to be here and see all of you. My group's work focuses on forests, which harbor astounding biodiversity and play a vital role in regulating Earth's climate. So as forests around the world are getting hit by climate change and a slew of other natural and anthropogenic pressures, it's very important to understand how they will be changing and what are the implications for their biodiversity and Earth's climate. Vital to global scientific efforts to monitor forests is the Forest Global Earth Observatory, which is led out of the Smithsonian and for which I lead the Ecosystems and Climate Research Program. We are the only ground-based forest monitoring network applying a standardized protocol to forests all over the world. We now have sites on all 6 forested continents, 84 sites in 29 countries with over 100 partner institutions. And echoing what Bill said about the importance of partners for conservation and for science as well, those partners around the world are absolutely vital to making all of this possible. We're tracking over 7 million trees representing more than 12,000 species, and we've accrued more than 15 million tree stem diameter measurements. So globally, this is an extremely important network for forest science. One of those plots, one of those plots is right here. We have a 25.6 hectare plot right on campus. And following the standardized ForestGEO protocol, we have mapped every stem greater than 1 centimeter diameter, all over 80,000 of them. We've mapped them to a grid, identified them to species, measured their diameter, and then every 5 years we come back and repeat the process. It's a lot of work, but very valuable to science, as you will see. We have a history of accomplishments to be proud of. We've completed 4 of these full major censuses on schedule every 5 years. We've been monitoring tree health and mortality annually for 12 years, subannual measurements of tree growth for 16 years. We have a more than 30-year running deer exclusion experiment led by Bill McShea. And just for research on this plot, we have over 67 publications. We've had more than 100 early career researchers do part of their training here. And one moment that really brought home to me our impact was a couple of years ago, a colleague who's not connected to the plot or ForestGEO wrote a commentary on a publication of ours in Nature and labeled this as an iconic research forest. So we've really managed to bring this forest up and put it on the map as a globally important research forest. And this is because we've done work here that has yielded transformative insights. So one example, which is the paper that got this commentary, led by my intern, Cameron Dow, we published this in Nature, showing that the early arrival of spring doesn't boost annual tree growth. So the prior expectation is that warming springs and associated earlier green up of the trees would boost annual tree growth and thereby forest carbon sequestration, resulting in, a mitigating effect on climate change. But we used dendrometer bands data from here, as well as Harvard Forest and tree ring records from here and across the eastern US, to show that growth is starting earlier in these as the springs warm, but it's also ending earlier. The whole process is moving forward. And there's no relationship between spring temperature and annual growth. Rather, the effect of warming temperatures comes in late spring and early summer, and it's a negative effect. So warming overall is reducing tree growth. And the implication is that these forests will not sequester as much carbon as expected as the climate warms. meaning we need to do more with reducing emissions rather than relying on forests as much as we had hoped. I see I'm out of time. I'm just gonna speed through I'll speed through these last two slides. Another project showed that rather counterintuitively in forests all over the world, it's the larger trees that suffer more during drought. And going into the future, we have exciting plans. At this plot, we're going to be looking into why we are starting to see biomass loss in this forest when the expectation is that these forests will be sequestering carbon into the future. And we're also involved in geotrees, which is a big global effort to calibrate it's a ground-based reference system for satellite-derived biomass mapping, which is essential for carbon trading and such. We have funding for the tropics, but not yet for temperate regions. And we've just made SCBI. We've done the measurements needed to be a geotrees site, and we're looking to lead with SCBI as one of the first temperate geotrees sites. So that's it. Thank you.
Thank you, Krista. Our next talk is Buddha with 2 Ds, Pukazhenthi. No one's ever spelled his last name properly, but he's going to talk on the artificial insemination of ungulates.
Good morning, everyone. I just want to start by saying, I think Brandy mentioned, everybody has a story and how you ended up here. I had a dream, it's true, 1985, back in India when I decided this is where I want to be. This is where I want to be part of this team. So I'm so honored to be standing in front of this group, sharing my experiences in this organization. And what I'm going to present here is also not just what I am talking about, and it's not me. It's a whole host of team Kevin talked about. It's a whole team that exists here that makes this possible, whether it's building infrastructure, training animals, making animals do certain things. As scientists, we come and kind of parasitize all of those skills and make science happen. That's what I'm going to try and do here. We already saw the history. This place used to hold hundreds of animals. And we've significantly reduced the collections for various reasons. I think the focus of science and all of it, the boxes that you see colored, those are the current species that I am leading research with the rest of the team here. So we have changed quite a bit over the years. What is unique about this? I call this place a very special place. There are a lot of unique things. We are, I would say, maybe one of 2 institutions globally that has the kind of infrastructure and expertise that exists here. Infrastructure, we have to build that infrastructure to handle animals safely. We are able to hold large collections of animals in our here. Experience of staff, we can never overlook that part. Research facilities that we've been able to invest and build those infrastructure, and this is for the long term. This is something that can exist hundreds of years if it's maintained well. So we are not talking about the lifespan of the species, we are talking generations to think about. This is sort of the general gist of what the research used to be, right? Most of veterinary medicine, nutrition, behavioral science, and reproduction is still a focus because the idea was to breed animals. We have changed where we evolved with the sciences. Now we are introducing all of the omics, genomics, and how do you use these kinds of advanced tools to manage these populations for the future. Reintroduction, we just heard Katherine's presentation about how we are advancing these sciences. We're able to do it because we're able to get our hands on these animals safely and repeatedly. How do you build a program for artificial insemination? These are the puzzle pieces, right? You have to understand the biology of the species. Talk about how do you collect sperm from a male? How do you preserve that material that you're handling? How do you assess the female? Then you look at how do you control the female reproductive cycle so you can do these procedures at a given time that is convenient for all of us. Monitoring, we have Janine Brown in this room, who is the mother of endocrinology, wildlife endocrinology. And then we put those pieces together to make that happen. This is a video that we made just to show how it is managed. Animals are conditioned to go through these chute systems on a regular basis, and we layer the science on top of it. So they come in, we have these restraint devices temporarily to hold these animals. Some of the collar work that we did was validated on this, where we were able to catch these animals, mark these animals, and look at how it's being done. We collect fecal samples to understand the reproductive hormone patterns. So when we we can control them. So I rely heavily on our staff. I am not monitoring these animals on a daily basis. They help us make sense. Collecting fecal samples, knowing which animal is what, where are we collecting the samples from? I cannot tell one scimitar from another, sorry, because 90% of the time I see the rear end of the animal, I don't see the front end of it. So here's samples being picked up. The fecal samples are being analyzed in the lab, and hundreds of samples we end up collecting for these projects, because we sometimes go back in time to see why something worked and why something didn't work. In the absence of this information, we are lost. It's really kind of shooting in the dark. Frozen sperm, I talked about the repository. We have been able to produce offspring with semen that was banked 20 years ago. Animal is dead and gone, but we are able to bring those genetics back into the population. So again, it's a team, right? It's just not me. I'm the spokesperson here, so I'll take the pride in that. We are able to work with the genetics and population managers who tell us what semen is compatible with each female. So pieces again, puzzle pieces, puzzle pieces, puzzle pieces. This is where we do the magic happen, where we do the artificial insemination. This animal is not anesthetized. Literally 15, 20, 30 seconds, we can do this procedure and the animal walks away safely and we are able to produce offspring. The animal walks away calmly, and the end result is, this is what we want to see. So what we have done over the years, we have been able to train a number of people. The idea is to replace yourself for the future. Graduate students, interns, postdoctoral fellows, visiting fellows, those are all people that we have been able to bring in here and also publish a lot of work. Some of the folks are here in this room too. Some of the firsts do matter. I think the point is, how do you apply the first? It's a tool. The tool is something that you develop and keep. You never know when you're going to need it. But 10 years from now, if somebody were to say, I need to reproduce this particular scimitar on oryx, I guarantee that we will be in a position to make it happen. So that's what I want to talk about. Thank you for your time.
Thank you, Budhan. So next talk is Andy Boyce-Piro from the Great Plains program in Montana.
First off, thank you everybody for your time and attention, and thank you for the invite to come and speak a little bit about what we're up to out in Montana, conserving grasslands in the Great Plains. So temperate grasslands worldwide are home to iconic species, from plains bison to Przewalski's horse to pronghorn antelope, like the one pictured here. But unfortunately, temperate grasslands are also the single most imperiled ecosystem on the planet. This is due to a combination of historical and contemporary rapid conversion to other land use types, as well as the fact that temperate grasslands are extremely poorly represented. I will. They're extremely poorly represented, both in our national and in our global network of protected areas. Temperate grasslands face a wide variety of threats. This is not just a historical problem. We're losing 1 million acres of grassland in the northern Great Plains in the United States right now. Beyond that, in the grasslands that still remain, keystone species like prairie dogs, bison, and important ecological forces like fire are almost entirely absent from these systems. This has created a crisis, not just for the biodiversity of temperate grasslands, but also for the people and cultures that rely on them. So with that threat looming, Smithsonian initiated the Great Plains Science Program under the leadership of Bill McShea in 2018. I'm ecstatic to say that I was one of the first people to join that program and move to Montana. Immediately, we found partnership with a couple organizations, American Prairie, which is a rewilding organization, and also the Fort Belknap Indian community in north central Montana. Both of these groups have both access and manage a huge amount of land and do so purely for biodiversity, or largely for biodiversity conservation, and in large part via restoration of native keystone species. I'm proud to say that there's far too much science for me to go through during this talk, so I'll try to just touch on a couple highlights here. Our group has done a tremendous job in increasing our understanding of how native keystone species interact with and support grassland ecosystems. We've been able to show that restoration of bison and prairie dogs increases the nutrient content of plants in grassland systems, and also increases the diversity and landscape scale heterogeneity of vegetation in grasslands. Beyond that, we've uncovered completely novel interactions between grassland keystone species and other members of the community. This illustration here is of a long-billed curlew in two different incubation postures on its nest. It's alert, relaxed posture, and it's sort of what I call its pancake cryptic posture. We've been able to learn through some really creative work by a graduate student, Andrew Dreeland, that these birds are actually able to eavesdrop on the predator calls given by prairie dogs and adopt these cryptic postures earlier with positive benefits for reproductive success. I want to shout out Nick Garnart, NZCBI's own Nick Garnart, who's an intern who created this illustration for us, which was absolutely phenomenal, and Ellie Thomasev, who did amazing work in comms that allows us to tell this story on NPR just a couple months ago. Beyond that, we do a lot of tracking work of imperiled grassland birds. These are tracks of long-billed curlews. Beyond revealing things like migratory connectivity and timing and links between the breeding and wintering grounds, we're able to apply this tracking data for direct conservation benefit. One specific example with these curlews is we partnered with an organization called Ovis in Mexico to do targeted long-term conservation easements on more than 10,000 acres of ephemeral wetlands in the Chihuahuan Desert of Mexico, where these birds spend the winter. I think one of our biggest and most significant conservation achievements as a program has been swift fox reintroduction to a large area of north central Montana where they were absent for more than 50 years. This restoration effort has been hugely successful with the translocation of over 150 individuals. These animals are reproducing, and as you can see in the figure here, they're also dispersing widely across this landscape, potentially linking up with a heretofore isolated population north of the border in Canada, which is very exciting. Smithsonian provided the scientific expertise and science support for this project, but ultimately this effort was led by the Ani and Nakota nations, the Fort Belknap Indian community. And for them, this work goes well beyond just a conservation success story. This was an act of cultural restoration for these people on this landscape, and we were honored to participate. As with any Smithsonian effort, education is a pillar of what we do. We train between 30 and 40 interns and graduate students every summer out in the Great Plains of Montana and North Dakota. This ranges from interns from the tribal colleges who are learning the newest field techniques to apply them on their homelands, to folks, young scientists coming to us from the global south and hopefully falling in love with grasslands and conservation for the first time. Going forward, we're really expanding our efforts into a wider variety of land use types and working with different people. We're now working with state agencies, federal agencies, lots of private landowners to provide science support for either rewilding, working lands conservation, or even restoration of degraded lands back to grasslands. One note I'll just leave you with, and I think it's an accomplishment or achievement that I'm particularly proud of, our team is proud of, is that through these partnerships built on trust and unbiased science, we don't own a single square foot of land in the Northern Great Plains, but our conservation science is now having an impact on upwards of 3 million acres across Montana, North Dakota, and other parts of the Great Plains. So thanks very much for your attention.
And while we're talking about the Great Plains program, I'll point out that Ellen Welty is here. This is her. She's a researcher out at the Montana program. This is her last week of work. She has taught us all the importance of dung beetles. And we really should give her a great shout out if you see her today. Okay, next talk is Sally Bornbusch, talking about microbiomes.
Hi, everyone. I want to reiterate all the thanks we've gotten. Thank you to the organizers and thank you for being here today as I talk about the really, really little things and how they can inform conservation. So what are microbiomes? I often ask the question to an audience, does anybody know what a microbiome is? I don't have to do that anymore. Most people know what microbiomes are. They are the communities of microorganisms that live within a specific habitat. And obviously, one of the first things that comes to mind for us is human microbiomes, right? The Human Microbiome Project was founded in 2007 and has provided incredible advancements to human health, understanding how those communities of microbes within humans, within every single one of you, all of you have microbiomes. How does that advance or kind of inform medicine? We now also apply microbiome science to things like agriculture. We understand soil microbiomes, how that influences crops and livestock. And we are now kind of moving into this era of knowing that microbiomes are critical components of human, animal, and environmental health. So how does that impact conservation? So far, we have kind of four main areas through which we're applying microbiome science to conservation. And that includes through augmenting animal health and welfare here at NZCBI and our collaborating zoos, combating diseases that threaten biodiversity, and forwarding our understanding of ecosystem function across the landscape, and finally, enhancing active conservation practices such as reintroductions. And what I kind of want you all to take away from this, if you take away one thing, is that microbiome research is a valuable tool that can be used to improve existing and future conservation strategies. So our first example, augmenting animal health at NZCBI, if you think about basically any animal that we have here on our SCBI campus or at the National Zoo, they also have microbiomes, and we can use that information, learning from their microbiomes, to improve how we take care of them and help keep them healthy. We have looked at things like gut health, digestion, metabolism, disease prevention, reproduction and development. I'll give you a super quick example. Just yesterday, I spent 4.5 hours processing 10.7 kilos of elephant feces into a fecal transplant for one of our elephants at the National Zoo who is having GI distress. So we're using our understanding of microbiomes to actually apply and integrate with animal care strategies. Also combating diseases, we have been using microbes to fight microbes. So we heard a lot about germs from Amanda's wonderful serenade, but we also know that not all microbes are bad, right? We have good microbes that can fight bad microbes. Examples include using microbes to combat fungal diseases like chytrid. So how can we understand amphibian skin microbiomes and their interaction with the pathogen? Using microbiomes in Hawaiian honeycreepers to understand resistance to avian malaria, and preventing things like GI infections in our black-footed ferrets. How can we prevent Clostridium or minimize the kind of impact of those diseases? We also use microbes at kind of a landscape or ecosystem scale. We work with our wonderful VWL partners to understand how microbes in soil influence these different regenerative agriculture processes and how that then translates into understanding livestock health and production. Finally, we use microbes to kind of enhance our conservation practices through rehabilitation and reintroduction. We've heard a lot of really great stories about reintroduction. One of the things we're highlighting is that we train animals in many ways to be reintroduced into the wild. Like Bill said, we don't just say, go free. But we now know that we also maybe need to think about how to prepare their microbiomes for life in the wild. How can we use that to help them prevent disease, prepare for new diets? All of these big questions. We have big projects with black-footed ferrets, improving reintroduction strategies using probiotics, fecal transplants. We also work closely with a lot of other animal facilities and conservation groups like the Marine Mammal Center in California to do things like improving rehabilitation of orphaned baby elephant seals, right? Like, that's amazing. For release, using microbiomes to improve that strategy. The future of microbiome science at NZCBI is looking really bright, right? We have a lot of wonderful new technology, a lot of knowledge. We want to continue advancing our animal care and health strategies. We also want to create kind of a hub to facilitate accessible gold standard microbiome science. This is a new field in conservation. We want to build international capacity for microbiome research and conservation. We have so much interest in this, and we want to build that capacity to train others in this kind of cutting edge new field. And finally, obviously, we want to train the next generation of conservation biologists to think about microbes and include microbiomes in conservation strategies moving forward. With that, I'll say thank you.
Thank you for all the speakers for staying on time. We're almost caught up. Okay, Molly Corder is next, talking about biobanking.
All right, hi everyone. So as many of you know, the Smithsonian Institution has been a global leader in biobanking cells and tissues for assisted reproduction and cryopreservation. And specifically, this has been done to support sustainable and genetically healthy populations of wildlife worldwide. And over the past 50 years, our scientists have banked over 100,000 samples from 125 different species. And now, as a complementary approach, our scientists are working to build a digital biobank. And the purpose here is to enhance health and reproductive physiology with previously unobtainable precision, using cutting-edge molecular medicine technologies that were first developed in cancer research. So how are we using digital biobanking to bring precision medicine to the forefront of endangered species conservation? Oops. All right, so over the years, our scientists have come up with multiple ways of assessing health and reproduction by identifying specific signals found within biological samples like poop or blood. And so each sample contains many layers of information like DNA, RNA, protein, and within those layers, we can find unique signals that can tell us about animal health. We can use that to understand animal stress or gut microbiomes or diets. And what I'm trying to say here is that these signals tell a story about animal health, reproduction, and well-being. And before signals can make their way into waste products like feces, they actually have to be present within circulation in the bloodstream. And blood is a treasure trove where we can find information, hundreds of thousands of signals, in fact, that are reflective of animal health. So we've done this right now in rhinos, looking at things like metabolites and immune cell proteins and DNA from their blood. So what happens when we start to integrate hundreds of thousands of signals from each of these biological layers? We can use multi-omic health profiling to create a map of a healthy, genetically viable animal. And these maps can be used to assess animal responses to stress or environmental change, as well as the likelihood of translocation or reproductive success. And we can start to ask the question, what makes animals resilient to health, reproductive, or environmental challenges? And can we then use these maps to enhance the welfare or animal care management plans of the animals that we take care of here at our facility? And we can also figure out how complex interactions between genetic and environmental factors are participating here and model these. So we're currently doing this with rhinos, looking at the epigenome to understand and discover how environmental influences shape the very expression of the information stored within their DNA. So now we can integrate these multiple profiles together, and I can introduce to you the digital twin. So this concept was first pioneered in human precision medicine, and it really is a map or a virtual replica, really, of the patient generated from multiple unique data sets on patient data, population data, as well as real-time updates on patient and/or environment or habitat that they live in. And so we can then take these signals from animals or the tissues and then use that to enhance conservation medicine. One of the biggest challenges that we face in conservation medicine is that we don't have controlled conditions to conduct clinical research. So enhancing species-specific or individual treatment plans is very challenging. So now we're starting to wonder, could we use these digital twins and artificial intelligence-based algorithms to leverage this patient data and then monitor, diagnose, and predict disease, and then hopefully go as far as to optimize treatment interventions. So we're moving past our typical electronic database deposits for clinical data and really starting to focus in on resilience and identify what are those features of shared health on our continually changing planet. And so as America's Attic, the Smithsonian is uniquely positioned to do this type of work. And we're doing a lot of other work in our programs to create a digital copy of our physical collections. And so we see this digital biobanking as an opportunity to digitally recreate our living collections. And importantly, these should also be linked to the physical tissue and cell samples that we store here on this campus in one of the largest wildlife biorepositories in the world, where these cells are waiting frozen in time to be used for conservation purposes related to disease pathway detection or assisted reproduction. And really, at its core, when we create these digital twins, we want to make sure that we maintain our leadership position as one of the major institutions who has paved the way for open science to exist and to democratize data generation as we develop data capital, such that access and global capacity building relative to scientific discovery is possible to assess individual and population health, guide sustainable breeding programs, and lastly, to enhance metapopulation management for species on the brink of extinction. Thank you.
Okay, the next is Autumn-Lynn Harrison, going to talk on migration.
Thank you, Bill. Good morning, everyone. You may also know me from the cover of the reports that were sitting on your chair this morning. So take a look at that. In 1970, the New York Times highlighted a then revolutionary study led by the Smithsonian and NASA. And they wrote, and I quote, "An elk named Monique and a space satellite named Nimbus have a rendezvous to keep later this week on a snow-covered plain in Wyoming. In an experiment to study the migration habits of wildlife, a Nimbus weather satellite 700 miles overhead is scheduled to tune in twice a day on the elk." Learn more about this text history on Lacey Huey's poster this afternoon at lunch. But since 1970, this technology's advancement from an 11,000 gram satellite collar to 1 gram satellite tags has directly paralleled our own growth at SCBI. Studying animal movement with tracking technology has really become an immutable part of our science DNA. 50 years ago, we could only guess where the animals we studied went when they disappeared from view, like this plover that I attached a transmitter to in Alaska. Today, we follow them across continents and oceans and into an uncertain future. Many shorebird populations have lost 70% or more of their abundance since 1970. The data you see here have been used in over 20 conservation projects. Being a part of the National Zoo, we use both the science, like a paper of mine, and the story, like this comic book illustrating the scientific paper, to connect with communities for conservation impact, aligning with our main Smithsonian mission, the increase and diffusion of knowledge. And now I'll highlight 3 stories of conservation impact that we have had from following footprints, flights, and flippers. We heard already from Katherine and from Andy about how they have tracked the first footprints of reintroduced fox and oryx into the wild after reintroduction, allowing us to assess the success of the initiatives. In Africa, recognizing recent population declines of the world's tallest animal, NZCBI scientists have launched a strategic partnership with the Giraffe Conservation Foundation, termed Twiga Tracker, and Twiga is Swahili for giraffe. This initiative aims to increase our understanding of the space use needs of all 4 species of giraffe and providing relevant information on the size of protected areas. At the Migratory Bird Center, we started an initiative funded by the Knobloch Family Foundation called the Shorebird Science and Conservation Collective. We've collated shorebird tracking data from scores of contributors across the Western Hemisphere. The brightest points on this map show where most of the data are found, and we then matchmake these data with conservation questions. In our first 3 years, we supported 38 total requests, and now we're up to 50. Some individual birds, of course, are the conservation superstars like Dr. Love and Dolly, the long-billed curlews, tracked by Andy, who have contributed to the most projects. Our tracking work at SCBI includes animals that also like to keep their feet wet. Suzanne Murray partnered with the Loggerhead Marine Life Center to track Falafel, an amputee turtle impacted by a fishing hook, to assess his resilience. My work has informed a new United Nations treaty for the ocean through a synthesis of global marine megafauna tracking. And we'll hear from Ximena next, who uses tracking data from sharks, rays, and marine birds in Peru to identify best practices. But taken together, our tracking studies across continents and oceans have created a living network of what Peter introduced this morning as wild data. The data you see here are from a project I lead, the Migratory Connectivity Project, founded by Pete Mara. Together, we have tracked 76 bird species at SCBI, and adding project locations for non-avian species, we cover the world. And when you look at a map like this, imagine your own families, your travels, your collaborators, your friends, distributed across the world, and how our science connects to all of them. And finally, Peter mentioned this morning that we have the opportunity to announce to you that the Smithsonian, together with our partners, have recently established the first global biodiversity observation network for animal movement, start and led right here to facilitate our data informing conservation at national and global scales. Today, I've highlighted the strength, leadership, and convening power SCBI has demonstrated over the last 50 years in translating our tracking science to conservation. And I really can't think of any other organization that has followed the footprints, flights, and flippers of a greater variety of species into conservation impact. So thank you for contributing to this.
Next talk is Alfonso Alonso. No. Oh, okay. Mina Velez talking on penguins in Peru.
Good morning, everybody, and thank you for having me here. It's a pleasure to be back in Front Royal and seeing some familiar faces. I'm a research associate with the Center for Conservation and Sustainability and the CCS under the leadership of Alfonso Alonso. We're working in four tropical countries, and today I want to share 4 short stories from our work in Peru, starting with penguins, specifically humboldt penguins. So in the central coast of Peru, 2 breakwaters that are part of a LNG port terminal unexpectedly turned into an amazing habitats for humboldt penguins, and this is a threatened species. From 0 penguins, this population grew to become the 5th largest colony in Peru in just 10 years. So this is a picture, like one of their daily pictures at the terminal. Abundant resources, basically little to no an interaction with humans, and ample and very suitable space made the penguin colony to thrive and even endure the double impact of avian flu and a very strong El Niño event a couple years ago. Along with the penguins came the boobies, the pelicans, the terns, and other species of birds, and turned the pile of rocks that we see at the terminal into a very lively community, and very loud as well. And as Autumn-Lynn was saying, we've been tracking the penguins for a year already and giving us a clue of where they're going, what they're eating, and another very useful information. So for my second story, I'm going to take you to the glaciers up in the Andes. Of the 180 protected areas in Peru, only 2 of these protect entire landscapes. The Noryauyos Cochas Landscape Reserve, you don't need to learn that name. It's in the Cañete River watershed. It starts at 5,000 meters, roughly 15,000 feet above sea level, and floats 200 kilometers into the Pacific. And this watershed serves 300,000 people. Here, the CCS is implementing research and conservation activities to foster water security using an approach that we implemented being part of the Working Land and Seascapes, reach to reef approach, meaning that from the glaciers that up in the Andes that provide the water, the fresh water to the river, and all the way down to the coastal areas where they finally finish. So we started doing what we do the best. We started looking into microinvertebrates, tiny animals. You barely see them, but they can reveal a lot about the water, the quality of the water. So the water, it's important. It's a precious resource for the communities, but also for tourism, for agriculture, and for aquaculture. And microinvertebrates can tell us a lot. How the communities changes and the richness changes tell us about the health of the river. And in this figure, you can see how the variation during the dry and the wet season up in the Andes. And with the blue, the best quality, and the red, the worst. And this is a very useful and powerful tool to communicate and also to guide management decisions. Following this project, we are working with Craig Fergus and amazing people in Peru, of course, my colleagues, integrating land use maps and hydrological modeling to build future scenarios of water quality and land use for the next 40 years. And we are co-building this with the local communities. And by doing this, we're bridging the gap of representation and also bringing their voices into the spaces where all the discussions or decisions are made. And for my final story, I'm going to take you to the desert area, where we're seeing how the El Niño events is changing the interactions in plants and animals and how it helps us our restoration efforts. So this is a desert area, and you can see the footprint there of a large underground pipeline. This is usually like the landscape that you're going to see most of the time, but when the heavy rains associated with El Niño comes, it turns into a vibrant community of plants, of flowers, and seeds, bringing abundant resources for hummingbirds there, birds as well, and mice and foxes, which are predators and seed dispersers. And we're monitoring these interactions to understand and help in our recovery efforts. So from the Pacific to the Andes, to the desert, these are the 4 encouraging stories that I wanted to tell you today, and that gives you a glimpse of our conservation efforts in Peru. Thank you.
Okay, the grand finale for our morning session is Amy Johnson. She's going to talk on Virginia working landscapes.
Okay, hi everybody. Thank you for your patience, and I'll try to make this quick, but I'm so excited that we're all here to celebrate the 50th anniversary, and at the same time, we're also celebrating 15 years of Virginia Working Landscapes. So I'm kind of going to go through a timeline of what we've been up to the last 15 years. But first, I just want to point out this incredible team. A lot of the success we've had is because of them. So if you see these faces around campus today and tomorrow, make sure you introduce yourselves because they play such an important role in everything that we're doing. So I want to take you right back to the beginning. In 2010, a group of landowners approached the Smithsonian and several other partners about trying to discuss how we can increase our knowledge of what's happening on private lands. Bill McShea really started this effort from the ecological standpoint. Ruth Stolk was here to help garner community support. Tom Wood was here at the time, was at the Environmental Studies on the Piedmont, which is now the Clifton Institute, but also local organizations like Piedmont Environmental Council, American Bird Conservancy were all at the table. And together, they came up with this concept of Virginia Working Landscapes with a mission to promote the conservation of native biodiversity and sustainable land management through scientific research, through community engagement, and through education. And what started then has become an incredible journey that I had no idea we'd get to where we are today. So Bill started the program with these foundational biodiversity surveys where local landowners would open up their gates of their farm and invite us in with a cohort of community scientists to study populations of birds, of soil, of plants, of pollinators, and from that develop more focal projects that can help us address questions about land management on private lands. So for example, this is Allison here, who's currently quantifying ecosystem services provided by cavity nesting birds on farms. We're looking at soil and cattle gut microbiomes with Sally that she mentioned earlier. We're looking at how to restore and manage our native grasslands. Those are just a few examples of what we're working on. But I want to point out that this program was started entirely through community support. So we had a group of core donors that really helped spearhead this program in the beginning, along with Bill and Tom and Ruth, and many of those supporters are still with us today. So over the last 15 years, our first year in 2010, we monitored about 10 farms that opened their gates to us, and to date, we've monitored over 300 farms throughout the 16-county region, covering about 95,000 acres of private land. And through this period, we've also built collaborations across more than 10 SI units, so bringing in Smithsonian expertise to help us bridge these gaps about land management, and then also integrating the knowledge of those local communities and co-designing that research together. We've built more than 50 community partnerships, trained more than 30 interns and fellows, including just yesterday, our PhD fellow, Bernadette Wrigley, became a doctor. So we're really proud of her and all her efforts. Led by Erin Shibley, we've trained over 150 community scientists to support our biodiversity surveys as well as our research. We've gone from about 5 donors to over 100 that they support our program entirely. And we're today now using this research to inform conservation at the state and national level. So for example, with the state, we are now providing incentives to farmers who are adopting biodiversity-friendly farming practices on their properties. So I want to point out, the heart of this program and what has brought all of this success is really the people and the partnerships, our alumni. I can't go on without acknowledging Dr. Tom Ockery, our founding director of Virginia Working Landscapes, who really set an incredible foundation for me when I came on board, but also all of the volunteers and interns and staff who have helped frame this program. The farmers and landowners who open their gates, they come out with our staff when we're out doing surveys, and they really share a lot of knowledge that ends up being built into our research design. And now Virginia Working Landscapes is seen as a model of collaboration for promoting community and science, not only in Virginia, not only in the US, but also on a global stage. And we're really proud of that. So I just want to end by saying, yes, we're celebrating 50 years here with SCBI. And for us, 15 years is a really big milestone for VWL. I'm really proud of all that we've been able to accomplish as a team, but I also really see this as a springboard for what's to come. I kind of feel like we're just getting started, especially seeing all of the potential collaborations that are gathered here today in the room. So together, we're going to continue to innovate at the intersection of wildlife, agriculture, and communities. And I challenge you, for everyone in the audience and everyone tuning in online, to think about what your role could be in community level conservation as we continue to really drive impactful conservation forward on working lands, not only just in Virginia, not only in North America, but around the world. So thank you so much.
So this concludes our morning session. I know I'm standing between you and lunch, but there's a few things I need to say. First, I want you to give an applause to Bill. Wait just a second. Bill actually this year received 2 major prizes, very well deserved career prizes. He has received the Aldo Leopold Award from the American Society of Mammalogists, recognizing his impact on conservation in North America. And he will be giving he received the Secretary's Prize for Scientists and will be giving a keynote speech on October 7th. So anyone that has time, join that talk where he will talk about his career accomplishments. So Bill is clearly one of our conservation giants, and thank you for doing this session. I'm also just blown away by the quality of the talks and the dedication and passion that our scientists showed just this morning. And so maybe that gives you a little bit of a hint why I talk about these big goals. To me, it seems like we have everything we need. We just need to package it better and move forward and putting it out there.